Hello there, friends. So we are finishing up our upper respiratory, lower respiratory tract infections. Just a joke there. And one of the things we were finishing up was the decision in the U.S. to not actually carry out the administration of a tuberculosis vaccine. And we'll be talking about vaccinations and vaccines later in the semester as well, and obviously this is something that's on a lot of people's minds. Once you've been vaccinated, it will look like you have been exposed to the pathogen. And so if you vaccinate lots of people, everybody is going to test positive for TB, if you're vaccinated for TB, for TB in this case. So in Europe, they do that. And if you were to, a European coming to the U.S. and decides you want to go work at the YWCA daycare and they're going to do a TB test, you're going to light it up like a son of a gun because you have antibodies against the bacterium. It looks like you have TB. Now, that means that you can't actually do surveillance, and I guess the general health notion is TB is not sufficient enough of a problem to actually start up a wide-scale vaccination. I mean, we get vaccinated for lots of stuff, and now COVID, we got one more thing. I would not be surprised if we start requiring this as a vaccination in order to start public school, because, you guys, when you started here, had to show that you had an MMR, measles, mumps, rubella, probably diphtheria, tetanus, pertussis vaccine, which, you got back in the last booster was in 5th grade or something, and it was a long time ago. So it's not out of the ordinary to require vaccination records. And I think it'll be just an interesting game is, do we require a COVID vaccination record? I personally am like, yeah, heck yeah, man. If I could lecture without my mask and know that everybody in the room is been vaccinated and we can kind of get back to working in groups, I'd do it. But anyway, so we finished up with this, the kinds of infections. And, pneumonia is the term that's used, but really it is a number of different bacteria. And consequently, if you've got a non-standard pneumonia that's not responding to the standard antibiotic treatment, they probably will go ahead and do a nasopharyngeal swab, stick it up your nose, stick it way back in your throat, and actually culture it and look to see. This level of bacteria culturing and identification, I would imagine that DMH is doing this pretty regularly. And probably, rather than going through the metabolic tests and stuff, they've got little antibody test strip kits that they can just go ahead and check very quickly to identify some of these basic ones. So walking pneumonia, double pneumonia, I used to think double pneumonia is like, twice as bad, which I guess it is, but what it refers to is both lungs. Walking pneumonia is, in fact, a bacterial infection. It's not so bad that you're laid up in bed or you're in the hospital. And the point to keep in mind is that if you go digging around in your nose and pharynx and lungs, you're going to find, for sure, Staph pneumoniae, Staph aureus. You might even find some of these other guys as well. You've got them right now. It's just a matter of how much of it do you have. So we end up having a range of tools that we can use for diagnostics. One is a chest x-ray, and you begin to show fluid in the lungs. And this isn't going to be exclusive to pneumonia, but there's other kinds of things you would look at as the sputum and things. A person that would have congestive heart failure probably would have it on both sides, and it probably actually does manifest itself a little bit different. At the ultrastructural or microscopic level, you actually have little spots in the alveoli that show like these thickened walls here. This is the alveolus collection. And also a bunch of the cells of the immune system, neutrophils, are present here along with macrophages. We'll be talking a little bit more about these guys in the immune system. And so a pathologist can actually look at this and say, I mean, I would imagine it's probably post-mortem. I don't think they do a lung biopsy on you. You then can have, at the gross level, this sort of necrotizing effect in which you actually have pretty significant lung damage. So now, there are a couple of interesting kinds of upper respiratory or lung infections that are non-standard. And part of this is a bacterium, Mycoplasma pneumoniae, that actually can have a really fast phenomenon in that they're very, very small. They don't have a cell wall. They are actually, Mycoplasma are oddball bacteria. They attach the cell to the cells of the cilia, and they paralyze them with a toxin. And you end up having a thickening and an inflammatory response, and you also end up having the inability to brush dust and particles out and mucus, and you start to have, filling things up. And it's one of the side effects of this as well, is if you can't brush the mucus out, you end up with the sticky stuff in your lungs, and that in turn begins to trap more bacteria, and it's just that much more of a mess. So kids with cystic fibrosis, they are building up very thick mucus in the lungs, and it makes them much more receptive to pneumonia. Another one thing you might have heard of, perhaps, it's been, it was identified, back in the 80s, maybe? No. Yeah, in the 80s. 76, sorry. In Las Vegas, I think. And a hotel that was hosting an American Legion meeting. And a number of individuals at this American Legion, which is like a fraternal organization, a number of individuals got really, really sick with a unknown bacterial infection, and several died. They later went back and identified there was mold and bacteria growing in the air conditioning system. You have the condensation pan, and it wasn't very clean. And they identified a previously unknown bacterium that is now known as Legionella pneumophilia, because it caused pneumonia in legionnaires. And this is an odd kind of bacterium in that it actually is growing inside the cell, and it infects parts of the immune system, the phagocytes. This is, in nature, is actually interacting with amoebas, which is how it's actually being maintained. And then it can get into animals and start having all kinds of difficulties by disrupting the cellular mechanisms and things in the lungs. So it is probably in some kind of biofilm, and one can imagine in sort of a drain pan of a air conditioning, you can end up with a little slimy layer of bacteria and stuff, and that's where this started. And it actually is a fairly significant pneumonia-causing bacterium, and a really oddball one too. All right, so our next step in the sort of march through the system, and to those of you in the audience and those of you in TV land, I got to look to see when our next exam is and what we're covering. I can't remember off the top of my head if our next exam stops at the respiratory tract or if we get into this one. I'm going to look and I'll get back to you. There you go. We'll do the next exam the same way we did the first one, where it'll be just an online thing or a take-home thing. So we're going to start with the next sort of entry point for a whole range of diseases, and this is in the gastrointestinal tract, i.e. your mouth. We can break these things down into a couple of different kinds of manifestations. I mean, one thing to think about is that the digestive tract is a pretty harsh spot. You got acids, you got all kinds of different hydrolases that are breaking things down in the stomach. I mean, most digestion takes place in the small intestine, but still, a lot of things that you eat, I mean, everything that you eat is covered in bacteria. You're probably destroying a lot of those bacteria when they hit the stomach. And those that you aren't destroying probably can't survive in the GI tract. And, if a healthy gastrointestinal tract is filled with bacteria, so, you eat your yogurt. But that said, we can break these down into bacterial infections versus this idea like intoxications, of where you have bacteria that are producing very potent toxins. And this is an example, as we'll see here. Ground meat occasionally will get recalled, and there are specific strains of E. coli, which is really a non-pathogenic kind of bacteria, but there are strains of them that produce toxins that can make you very sick pretty darn fast. So, starting out with one of the top guys you probably have heard of, the bacterium that causes botulism, Clostridium botulinum. And it has some interesting kinds of mechanisms in the way it works, in that it's an obligate anaerobe. And so if you've got a canned food that hasn't been processed properly, and I always think this is kind of an amusing topic to bring up, in late fall, when people are starting to can stuff up, particularly around Thanksgiving, and this is right about when this is coming up, pumpkin and different kinds of dense vegetables like that, the canning or the home canning industry says don't do that because it's neutral pH, it's very dense, and it's difficult to make sure you have heat and sterilizing heat all the way through that dense material. So the guys at the Libby pumpkin plant, they've got test cans that have temperature probes stuck in them, and so put them into their autoclave, and they're making sure that those test can interiors are getting the appropriate temperature. Because what can happen is if you don't hit it at the appropriate temperature and kill everything off, it's not that botulism can grow, but what will grow immediately is a whole range of bacteria that are present just on the surface, on the machines and the air, whatever. They begin growing and consuming what little oxygen is in that jar. Once they consume the oxygen, it's now gone anaerobic, and that's when botulism can take over. Because if you maintain an aerobic environment, botulism isn't going to do anything. It can't grow. But if you generate an anaerobic environment, that's why we've got actually a container that you can generate an anaerobic environment in. And other ways I've seen doing it is like taking a PVC pipe that you can seal up on both ends. You put a stack of plates in there, and you take a candle and light it, close the lid, and the candle, as it burns, consumes the oxygen, and eventually it's going to burn itself out because there's no more oxygen. You can generate an anaerobic environment that way, and from the soil that we picked up, you could culture some really nasty stuff from that. There's also like chemical packs you activate, and very expensive anaerobic environment, incubators and stuff. But A, we don't have one. B, I ain't interested in growing anything that nasty. But botulism, you can do. It's gram-positive. It's pretty easy to recognize because it has an endospore. It has a little ball inside it. And it is a fairly rare kind of thing. So that's good. The number of cases is usually in the U.S. under 20, but there are occasional outbreaks. So a bulging can lid, and this is why, one of the reasons why, if you have a can that's got a bulge in it, probably not good. If you've got one of those jars that has a seal on it, with a lid, but the top has been bulged, again, that indicates it's either been opened or there's gas-producing bacteria in there that didn't get killed. Of course, we know of botulism from Botox. The botulism toxin blocks the receptor on muscles that responds to nervous system signals. And so it means that you cannot contract and send a signal to those muscle cells. Consequently, it causes muscles to no longer contract, and it will relax the muscles in your forehead and relieve you of your pesky muscle wrinkle, the wrinkles in your forehead. And you can do the same thing with different parts of your face, all kinds of parts of your body. You can block the nervous system signals getting to the muscle cells. It's not permanent, but if you do it enough, it's probably not good. So there are a number of different kinds of botulism associated. The poisoning by eating it from different kinds of foods. Infantile, typically it's found when babies are fed honey. Little babies really shouldn't be given honey because their immune system is not able to adapt to it. And particularly raw honey is not pasteurized, and it has a range of potentially dangerous spores and things. And just not to belabor, but honey's got a lot of sugar. And so osmotically, cells can't survive or thrive in it, but spores can, because it pulls the water out of everything. Once you're old enough, raw honey is probably a fairly good food in terms of exposing you to different pollens in your area and stuff like this. But for little babies, probably not. And a lot of honey these days is coming from overseas, shall we say. It is not particularly naturally occurring honey. I mean, it's everything from corn syrup that's been flavored and colored to honey that's been highly processed. And the reason or the health benefit of honey is that if it's wild from the area where you are, it contains a variety of pollens and allergens, and it can actually serve as the equivalent of like a little allergy shot. Regardless, babies show a decreased muscle tone as the botulism begins to block the nervous system signals, and you start to lose muscle tone. Wound associated, you can get spots deep inside. With food poisoning,
we'll get onto that. But this is, as the building name implies, is the
so neutrally pH foods that don't get canned real well, you're going to have bacteria present and not just the Clostridium. So it's a really good idea to keep your cooked foods hot and your cold foods cold. We'll be talking about more food poisoning type systems. Symptoms, you start to have muscle difficulties. Mainly, you start to see it from droopy eyelids and the difficulties in standard muscle contractions like breathing and speaking. And if it's untreated, you bet it's fatal. So let that be a lesson to us. It is considered one of the most deadly toxins known. And what it's doing is blocking the release of neurotransmitter. And not that we have to go into too much of the cell biology of this, but maybe in anatomy and physiology, you know about the neuromuscular junction in which you have an axon of a neuron, the tip, is right next to a muscle cell in this gap called the synapse. We say it's synaptic to the muscle cell. And this gap is where neurotransmitters are released. And neurotransmitters are being carried by these synaptic vesicles, and what we refer to as the presynaptic neuron and the postsynaptic muscle cell. Well, botulism is actually working to inhibit the binding of the vessels to the cell membrane and preventing the dumping of the acetylcholine. So I'm just thinking about this. One of the treatments potentially could be nicotine, but that's a very different story. Nicotine affects the enzyme that clears the synapse of acetylcholine, and it keeps the signal there longer. Too much nicotine actually results in is a pretty potent poison as well. So I don't know that I ever hear of botulism being treated with nicotine or either one of them being used back and forth, but it's an idea. Anyway, it is a very potent neurotoxin. Okay, now the most common source of food poisoning is probably from Staph aureus, and we've talked a little bit about this guy. This is probably on everybody's skin at any one point. We are covered in Staph aureus. And this again is the idea that it's really not just the bacteria, it's the amount of bacteria. And I think we can kind of make a distinction, is that most food poisonings, botulism being the rare exception, where a little bit of it is very deadly, most food poisonings occur from bacterial infections in the food that reach a certain, what I think of as the bingo point, as the diarrhea point, that you don't really need to have your food sterile. And if it's coming fresh out of the oven, it is sterile. Now, it may not be sanitary, but it is sterile. And the load of the bacteria is really critical. And so if the bacterial load stays low, even though you've got Staph aureus on there, and you do, the amount of it is low enough, you don't have any difficulty. But if you let something that has Staph aureus in it sit at room temperature, or even better, 37 degrees, and let it go for 4 or 5 hours, you can begin to generate enough, that you give yourself food poisoning. And so a classic way of thinking about this is picnic season. So not, the whole notion is you don't want to have your deli platter or potato salad that you set out on the picnic table and you let it sit for a while. And we're going to go off and play volleyball. And then we're going to come back and have potato salad. And then, let's go swimming. And I'd like to have maybe another sandwich. And this stuff has been sitting out. And so over a period of 4 or 5 hours, the amount of bacteria gets to a spot where you're not going to be happy. And so the protein or the bacteria produce a toxin that, if you've got enough of the bacteria, it's going to make you sick. So it's gram-positive. It's not modal. It doesn't move around. And it is present on us right now. This is the bacterium that is responsible for the methicillin-resistant Staph aureus forms. And that's a problem because if you get too much of it, you can't really knock it back because it's resistant to most antibiotics. Absolutely, if you were to take a nasal swab in the room here, heck, probably swabbing surfaces, swabbing skin, you're going to find Staph aureus all over the place. So that's why you shouldn't lick the desk. All right, so the point being that the food comes pre-contaminated. You ain't got to add it, it's there. Being more careful with your food is important, certainly washing your hands. But we end up with stuff that is maybe being held without proper chilling. That's a great place. The toxin is heat stable. And that means that if you have enough of a load, you can still get it. But I dare say, if you've got enough of a bacterial load on a piece of meat, God knows what you'd have, you're going to look at it and you're going to go, man, that smells dank. I am not eating that, even if I'm going to cook it. Which gets me back to my general nutrition model, and I'm not entirely sure if it's true, but I still live with it, is that if you could eat it cooked, fresh out of the oven, and it didn't make you sick, you probably could have eaten it raw, fresh out of the refrigerator. I ain't eating raw meat much, but it's like we can get overly nervous about it, but most of these things are heat stable anyway, which meant that if there was enough bacterial toxin and bacteria in that food to begin with, cooking it didn't make any difference. The other side of it, though, is if you let it sit, there you go. So salads, fresh cream cakes, oh yeah, milk and dairy products, all these different kinds of things that we're leaving out are going to be a problem. So it's being passed from people to foods. foods particularly that require a lot of handling, like the deli meats where they're stacking them up, that's a great place to do it. So the things that are prepared without cooking is not going to be knocking back the load, but it is reducing or it's kind of keeping the load down wherever it was before you put it in the oven. If you leave it out and keep it cool, or not at refrigerated temperatures, but not at oven temperatures, you're back to problems as well. Treatment, there you go. It is, as we say, self-limiting in that it's not going to be maintaining itself very well at the high levels in your gut. And so as long as you're not continuing to contaminate yourself, you get over it after a day or so. But if you haven't had food poisoning, well. All right, so now another Clostridium, remember there's Clostridium botulinum, is Clostridium perfringens. And this is a different kind of food poisoning. It is also gram-positive. It has endospores. It is anaerobic. It's bacillus. And I mean, not all bacilli are anaerobic, but this one is. All right. So you end up with a window in which you eat the food and you have a while before you begin to actually have onset of the disease. And this is often, again, things like cooked meat that has not been chilled. And because these spores are pretty darn tough, they actually can resist certain levels of heat and then actually germinate, begin to sporulate, and start dividing. And so that's how you can end up with this material making you sick, and why it would take a while for the germination to actually occur. All right, so those are our toxins. And generally with these guys, with a toxin, you just wait it out. I mean, botulism is bad news, and you may not be able to wait it out, but you're not generating an infection. You're eating this material, and another point of this is that if you cooked it, you can still get sick if it has sufficient number. The other side of this would be things that actually can begin to grow in you with different infections. And here we've got salmonella. So this is another standard food-borne bacterial illness. Salmonella enterica is responsible for typhoid fever. And, it is primarily found in areas that have poor sanitation. So it's gram-negative. It is actually able to move around, swim about. They don't tend to form some kind of biofilms and things, but it's found in areas that have poor sanitation, not very good public water systems, and it is the same old story of infected feces somehow getting into the oral tract. So people have it, and then they begin having diarrhea, and this is a way you can begin spreading this. Because if you remember, at the beginning of our, sort of disease systems, we talk about the fact that one of the things that's going to make a really good disease, good being for the bacterium or for the infectious agent, is that you have a way of spreading it. And so with the upper respiratory tract, we have coughing. With the GI tract, it's primarily good old diarrhea. Vomiting doesn't do you nearly as good as diarrhea does. So the bacterium is irritating the lower intestines, and whoop, out we go. All right, so this can be a bit more serious in that the bacteria that are going to be responsible for these kinds of things, in addition to salmonella but other types, they typically are acid resistant. So you eat them, and they're actually able to survive the stomach transit, and from there they can get into other parts of the body. Now, a couple of things to think about is that if you are in pretty good health, it's not going to be as severe. But we can have small perforations within the GI tract. And things like, bones or different kinds of rough foods might put little perforations in. And most of the time, it's not going to be a problem. But you can end up with small tears and perforations that can allow this bacterium to now get into the peritoneum, and you now have a much more serious problem. So the enterotoxin itself that the bacteria generate is able to actually begin eroding parts of the epithelium of the intestine. And so you have bloody diarrhea, oh boy. From there, the bacteria can start getting into the bloodstream. So if it goes long enough, a systemic infection, you can generate a really high fever and a pretty serious mortality rate. It can take a while for this infection to actually be knocked back. And so one of the points to make here is we've got now the bacterium actually culturing within the body as opposed to not being able to make it but having generated a toxin that causes you to have difficulties. This actually is the bacteria growing in you. So you perhaps have heard of Typhoid Mary, and one of the things that can happen is you can be a carrier for this bacterium in that you don't have particular symptoms, but you're actually transmitting it and passing it on. And so this individual had the bacterium, was either in a subclinical case or somehow resistant, and was actually contaminating members of the household that she was cooking for. All right, she actually was cooking then in 1915 for a hospital. Not great, because she had a long-term infection. Returned to quarantine for life. So she had a bacterial infection in her gallbladder that was constantly dumping out small amounts of this bacteria, probably, potentially through the feces and then washing your hands after the toilet. In 1900, how much sanitation was being done. It's why, if you go into a restaurant, you see the little sign in the bathroom that says, all employees must wash hands before returning to work. Well, I think everybody should wash their hands before returning to work, but particularly if you've taken a poop and you're about to go back into food handling, wash your hands really damn well. Just as a little thought there, a public safety announcement, because that would actually have prevented a lot of this problem. But if you're a carrier, you got this. All right, so Shigella is another kind of bacterium that is responsible for dysentery. And dysentery is one of these kinds of terms we use for rampant diarrhea. And there's amoebic dysentery, there's bacterial-induced dysentery. So shigellosis is one of the terms for this. Poor water sanitation, again. And you have unwashed hands, the oral pathway, going through the stomach into the intestine. You get bloody diarrhea. You dump it out. The pathogen is in the stool. It can then get back into the system, in a sense. Flying cockroaches, spreading the disease. You then get into more food, you get into water, and it can become a really unpleasant cycle pathway. That's again one of the reasons why the health department checks out restaurants, and you want to limit the amount of roach infestation. You don't need those guys tracking things from one place to another. Thank you. All right, so it is a typical oral fecal route. Again, treatment is antibiotics and hydration, and you just hold on and let the guy clear it. So it's invading intestinal cells, actually getting inside, and it is causing harm to the epithelial cells of the intestine, and that in turn limits their ability to pull water out, and you have sort of an inflammation kind of like a Crohn's type thing, where you just start dumping. Pleasant. Cholera, you perhaps have heard of. This is a gram-negative, is motile. It's actually got a little bit of a curve to it, and so it's pretty easy to actually identify in the microscope. Poor sanitation, and again, having a good sanitary sewer and a good water treatment plant goes a long way to keep you from having these things. So very often it happens, you get onset is really sudden, and known as rice water stools. Oh, pleasant. You're essentially shedding the interior lining of the intestines. So you're going to have a bit of a long, hard recovery from this. Death through dehydration is the obvious thing. Your electrolytes get completely thrown out. If you can keep somebody hydrated, and probably through IV, after a time they actually can recover from it. But like they have specialized cots, beds, where you're lying on your back, and there's a little hole here, and they have a bucket underneath. I can't imagine a more pleasant way to spend your time in the tropics. All right, so the whole term rice water stools is not one of my favorite kinds of things. So cholera, actually we see it coming and going occasionally, and there was actually, more recently than this, cases of cholera, again, in some of the Caribbean Isles after either a bad hurricane or it was another earthquake. In Haiti, we have a really good example now of cholera coming up, and fatalities stay, more or less stable, although almost 1,000 people dying within 2 weeks, but the cases start to go up. And this is happening because you have poor sanitation and post-disaster conditions. Depending, you can have water main breaks. I mean, this is something that was a potential in Texas when they have. That's why you issue a boil water. There's the possibility of having cross-contamination between the sanitary sewers and the drinking water. These pipes are running near each other, and so you do have the possibility of having the water being contaminated. Not to get into too much detail, but cholera toxin is invading the intestinal epithelial cells, and one of the things it's doing is it's blocking the system that is involved in maintaining the appropriate salt concentration. So, we talked a little bit about osmosis, and you probably talked about this in A&P as well, but you don't pump water around, you pump salts around, and water will follow salt. So when you have your food in your stomach is then squirted into the small intestine in small doses, and the small intestine begins pulling out all the nutrients and things. The large intestine's job is pretty much nothing but pulling the water out to give you a nice firm turd. Now, if the way you pull the water out is by pulling the salt out of the food, so you begin taking up the chloride and the sodium ions out of the poop to be, and water follows salt. So if you pull the salt out of the food, the water comes right along with it. And you then have a nice firm stool. So one thing is, if you ingest a large amount of an ion that we don't pump out real well, say magnesium, you end up pumping out the sodium and the chlorine, but you still have all this magnesium, and it's pulling the water back into your poop to be, and now whoosh. And so for colon prep, that's what you do, is you take a big magnesium bolus, and they even offer and sell things like milk and magnesia, magnesium-based laxatives. They work because magnesium isn't pulled out very well, and you're just adding this ion that is osmotically keeping the water from being pulled out. Well, in the case of cholera, you're blocking the chloride transporters and things, and you're changing the way the cells are able to pump salt out, and consequently, they're not able to change the salt concentration such that you have lower salt out here, higher salt here, pulling water in, whoosh. You end up essentially blowing out unprocessed poop. All right, so now another bacterium that if you're a ground meat fan and you keep an eye on this, we have occasional ground meat recalls. I think it said that like when they do a recall of ground meat, they will get less than 20% of the ground beef back. Part of what this means is that while the contamination may be there, it's not super terrible. People survive it. But regardless, E. coli. And we know E. coli as a gram-negative little bacillus. We use E. coli in the lab all the time, and it's our standard cloning bacteria. And it is possible to be carrying a plasmid that produces a range of antibiotic and resistance genes, and additionally some toxins. And so most gram-negative, most E. coli are non-pathogenic. I mean, we got flasks of E. coli upstairs that, other than the fact that it smells a little bit like dog poop, you could drink it and it'd be as beneficial for you as yogurt. But there are some E. coli that do pick up these plasmids, and now they can cause some illness. And this happens because E. coli is a bacteria that is massively found in your poop and in the cow's poop. During the butchering process, the cow is knocked in the head, falls over. They very quickly begin to dress it, to begin dissecting it apart. And one of the first things they do is they pull out the intestines. In the operation, there is going to be a little bit of contamination. There just is. And if you have the bacterium with this specific contamination, you can end up having meat that's contaminated this way. And it's one of the things we think about ground beef, is that if you've got a bacterial infection that's on the surface of your steak, when you sear that guy, you're killing the bacteria. And unless this cow had a systemic infection, which is a pretty screwed up cow, the inside of that piece of steak really should be all cow. It should be all sterile, except for cow meat. So the surface is contaminated potentially from the cutting and processing. But you sear that. All right. Now, when you take ground meat, you're taking a chunk of meat and grinding it up, and effectively the outside gets mixed in with the inside, and consequently you don't have this sort of separation of potential contamination on the surface, but it's all clean inside. It is potentially contaminated throughout. Consequently, if you're a germaphobe and really nervous, a rare hamburger is not something you should ever eat. On the other hand, if it's clean and reasonably prepared, it's probably okay. But I don't eat. I've never eaten raw hamburger. Apparently, there's a thing in Wisconsin, and like this past couple of weeks, it was some sort of raw meat season, and I don't know. Like you eat raw hamburger meat. I'm from Texas, man, and we don't do that. But regardless, so occasionally we will hear about batches of ground beef being recalled, and it's because we expect a level of E. coli, but if there's specific E. coli like 0157, that's a standard one that is toxic. Now back probably 30 years ago, there was a little 4-year-old, I think girl, who died from contaminated ground beef found in a Jack in the Box hamburger. I'm not even sure if Jack in the Box exists anymore, but nobody likes to kill little 4-year-olds with their Happy Meal. And so from that point, the ground beef industry has been much more thoughtful about checking this. Do you recall from, now it's been probably 5 years, that there was maybe longer than that, spinach, fresh spinach was being recalled. And for a little window, you couldn't get fresh spinach at the grocery store. It was all frozen. And it's because the spinach was contaminated with O1570 E. coli. It turned out that the facility that was washing spinach in California, where the bulk of the spinach was being produced, actually had a cross-contamination with material from a cattle feedlot. And so all of the spinach was being washed in a gigantic toilet bowl and generating infections. Not so good. All right, we're going to knock off here. We're going to pick up with gastric ulcers and actually some cool ideas about how gastric ulcers form and continue from there. So, that's all I know. And I got post stuff. Ciao. I'll see you guys.